Good morning, everyone. Hope you're well. This is not an April Fool's joke on the screen this morning. This is an article about actual Xenobots. As I promised you in this course, we're going to touch on a lot of different topics. We've talked a fair bit about robotics, engineering. We've done a little bit of math, some physics. We will do some chemistry. We've done a fair bit of biology. Wanted to talk for a few minutes this morning, just a few minutes about medicine. At the end of this course, we're going to talk about the Xenobots, which is evolving robots not made from metals and ceramics and plastics and electronics, but instead evolving robots that are made from biological cells. And over the last year and a half, a former member of this class, Sam Kriegman, with our colleagues at Tufts, has been able to run an evolutionary algorithm on the supercomputer here at UVM that runs an evolutionary algorithm using simulated frog skin cells and simulated frog heart muscle cells. The best evolved design that produces a simulated Xenobot that walks along the bottom of a petri dish. We send that to our colleagues at Tufts, and the microsurgeon there builds up from the blueprint a physical Xenobot cell by cell, and we've managed to actually cross the simulation to reality gap, which is one of the big challenges in evolutionary robotics, and we might actually start talking about that today or possibly tomorrow. So what can you do with a robot made out of frog cells? At the moment, not a lot, but as we move forward with this technology, it's possible that we can similarly evolve robots built from different kinds of cell types other than frog skin and frog heart muscle. It's possible that we might be able to evolve robots made from cells taken from other species, including possibly humans. And if we can build human biobots, there might be medical applications for this work. So in this particular article that covered our work on Xenobots, there was a fair bit of talk about medicine that we might be able to solve in the long term. We might be able to solve some of the major challenges in medicine by allowing human biobots to work safely and non-invasively inside the human body. It's a long way off, but that's the vision we're heading towards. We published our first paper on Xenobots over a year ago, and yesterday we published our second paper. I'll put up a link to it somewhere here on Teams today, and you can have a look if you're interested. But otherwise, we'll come back and talk about Xenobots at the end of the course. Okay, back to us, where we are and where we're going. We've started in on our theme of challenges in the field of evolutionary robotics. What are some of the big questions that remain to be answered? We're going to look at a number of those questions, and we're going to look at different answers to that problem. The biggest open problem, as you can see here, is the crossing the reality gap. We're going to look at 1, 2, 3, 4, 5, 6 solutions that have been posed in the literature so far. We started in on our second open challenge last time, which is the competing conventions problem. And we'll talk today about a potential solution to that problem, the NEAT algorithm, and then the HyperNEAT algorithm. Before we do, just a reminder that you're working on assignment 9, which is your parallel hill climber. It's the longest of all the 10 assignments, so please set aside enough time to work on that. Any questions about the assignments? Anything we've covered? Anything else? We're all good so far? Okay, so back to lecture 14. We ended last time by introducing the competing conventions problem, and I asked you to think about taking these two neural networks. Imagine we have these two neural networks that exist in the same evolving population. They're both okay. They both have, let's say, medium fitness. The first neural network is doing pretty well at computing subfunction A and B, but not so well at C. The second neural network is pretty good at computing subfunction C, but not so good at computing function A and B. It would be great if we could cut these two networks together and bring the good A and B from parent 1 together with the good C from parent 2. But as you can probably see now, if I restrict you to a straight line, and you have to use that straight line to cut through both networks in the same place, it is impossible to combine those together. One way to write this down or think about this is we have network ABC in the top row here, network CBA in the bottom row here, and we can, for example, cut here in network 1, which will give us AB in the first child. We get this piece in the first child. And if we're cutting here, that means we're also cutting over here in parent 2. So we take the A from parent 2 and we tack it on to child 1. Child has 2 copies of A and B, no C, so that's not so great. And child 2, if we're still cutting here and here, child 2 gets C and B from parent 1 over here, and C from parent 1 over here. Both are missing information, so it's hard to combine the best of both worlds. So we haven't talked about this too much yet, but in evolutionary algorithms, there are usually two variation operators. These are the algorithms that modify an existing genome in the population. You've implemented mutation, which is one of the 2 variation operators, and the other important one is obviously sexual recombination or genetic recombination, combining and recombining genetic material from 2 parents. The reason you're not implementing recombination in your parallel hill climber and in most other evolutionary algorithms is it doesn't work very well. It's very hard for evolution to blindly combine material from 2 different parents. Okay, so we're going to look today at the NEAT algorithm, which incorporates crossover and crossover recombination and increases the chance that a random recombination event will combine good genetic material from both parents. It sometimes bring together the best of both worlds. Okay, let's talk about the NEAT algorithm first, and then we'll see how it's able to improve recombination. Here's a visualization of what a single genotype looks like in the NEAT algorithm. Remember, the genotype is the blueprint of whatever it is we're trying to evolve. And in NEAT, we're going to be evolving neural networks. So the neural network is the phenotype. The phenotype is the thing that's produced by the genotype, by the blueprint. Inside this genome, the genome is made up in NEAT of 2 lists. So in a starting population in NEAT, you have a population of list pairs. The first list is a list of genes where each gene corresponds to a neuron. The second list, each node in that list corresponds to a synapse. So we've got 5 neurons in this case and 6 synapses in the list down here. If you look at the phenotype produced by this genotype, you should see 1, 2, 3, 4, 5 nodes and 1, 2, 3, 4, 5 synapses. One of the fields, or one of the fields attached to the synapses is whether the synapse is enabled or disabled. So in this case, we have 6 synapses, but one of them has been disabled. That's why we only see 5 synapses in the phenotype. Okay, each node in the neuron list has one label associated with it, and it's a label that indicates what type of neuron it is. In the second list, as you can see, there's 1, 2, 3, 4, 5 fields or 5 features that describe each synapse in the neural network. The first 2 are integers that indicate which is the presynaptic neuron and which is the postsynaptic neuron. So you can see this first synapse says, I connect neuron 1 to neuron 4, and sure enough, there's a synapse that goes from neuron 1 to neuron 4. There's the synaptic weight, which you're all familiar with by now. And then this Boolean flag enabled or disabled. And a 5th one, which is called the innovation number. And we're going to come back to what the innovation number is in a moment. This is the part of the genome that makes crossover possible. Okay. Okay, so that's the genotype, phenotype. Any questions? So far, so good. Oh, one thing I wanted to note here. There are a couple things that are new in the NEAT algorithm that we haven't seen before. Up until now, all of the evolutionary algorithms we've looked at, we, as the investigators, set the neural architecture of the neural network. We determine how many neurons there are and how they are wired up. And evolution is only free to alter the synaptic weights. In this case, we are expanding the evolutionary algorithm so that it can also modify the cognitive architecture. Mutations to this genome, if a mutation hits the neuron list, mutations can add or remove or change the type of neurons. Mutations that hit the synapse list can add or remove or modify synapses. So cognitive architecture is the number and connectivity of neurons in the network. As we continue through this latter half of the course, we're going to see how evolutionary algorithms have been expanded to automatically tune or tinker with more and more parts of the neural network controlling the robot and the robot's body itself. Okay, so let's have a look at NEAT, and let's see what happens during evolution when variation operators occur, like mutation. So there are a number of different kinds. There are a number of different kinds of mutations that can occur. For example, a mutation might add a connection to the synapse list. So this is now a picture of just the synapse list. There is also the neuron list, which is not being shown on this slide. We have 1, 2, 3, 4 enabled synapses and 1 disabled synapse. And you'll notice this particular genome has been mutated to produce this genome, and the mutation has added a new synapse at the end here. It's a synapse that connects neuron 3 to neuron 4, represented by the red line here. The numbers at the top, the integers at the top, these are the innovation numbers, which again we'll come back and talk about in a moment. Okay, here is exactly the same genome down here and the same phenotype. And in this case, we're thinking about this genome being hit by a different mutation, which in this case doesn't just add a connection or add a synapse, but it adds a node, or it adds a neuron. Again, the neuron list isn't being shown here. We're just showing the effect on the synapses. So in this case, we have a synapse going from 3 to 5, which we see here. We'll notice that this mutation disabled that synapse and added in 2 new synapses that go from 3 to 6 and 6 to 5, where 6 is the new neuron. So the authors probably should have shown this in the paper. There's a neuron list up here. The mutation added a new neuron, neuron 6, and then they added in a few synapses to the mutation operator added in 2 synapses to connect that neuron in with the rest of the network. So we now have, at the moment in your assignments, you have 1 type of mutation operator. That mutation operator finds a synapse and changes the weight of that synapse. In the NEAT algorithm, there are multiple possible mutation events: add, remove, or modify a neuron, add, remove, or modify a synapse. During evolution, which mutation operator is applied? We pick a random number. 0, we modify the neuron. 1, we modify the synapse. If we're modifying the neuron, we can choose another random number again. 0, delete a neuron. 1, add a neuron. 2, modify a neuron. If the mutation event we chose is to mutate a synapse, again, we pick a random number. 0 is delete a synapse. 1 is add a synapse. 2 is modify a synapse. So it's still random. But now we're choosing what mutation operator to apply, and what mutation operator to apply and how to apply it. Okay, so now we come to what NEAT was designed for, which is to allow for crossover. NEAT makes crossover, first of all, makes crossover easy, algorithmically simple. I'm going to describe how crossover occurs in a moment. And most importantly, crossover is designed to try and solve the competing conventions problem by producing children that are similar to their parents. And I'm going to talk about what I mean by similar. Intuitively speaking, you can imagine that if you're combining genetic material from two parents and the child is completely different from either parent, there is a low probability that that child is going to be fit or survive at all. Just to build up an intuition for this, look at these two parents here. These two parents are similar in the sense that they're both on their way to computing the three subfunctions required for whatever this task is. We haven't really specified what the task is. But you can see that these crossover events produce children that are different from both parents. Neither children have all of the three necessary building blocks. That's usually a bad thing. Okay. So how did biology solve this problem? So crossover or sexual recombination evolved in nature because, again, it's useful to genetically recombine material from more than one parent in the hopes that sometimes that recombination brings together the best of both worlds. But we don't want to bring in too many copies from one parent and not enough from another. So for those of you that remember your high school biology, chromosomes have alleles, meaning that for every gene, there's two copies. There's one allele and then another one. And during sexual recombination, we get one from each of our parents. We get one allele from each parent, right? We march down the chromosomes from mother and father, and at each point we decide for each gene, do I take a copy from mother or a copy from father? That is an extreme oversimplification of how crossover occurs in biology, but the intuition is we want to make sure that we don't miss anything or get too many copies of anything else. Okay. How does NEAT accomplish this? The nice thing about NEAT is, although it is encoding a network of neurons and edges, these networks are represented as lists. Here's parent 1 or network 1, here's parent 2 or network 2, and both of these have a list representation. Again, in this cartoon, we are not showing the neuron list, just the synapse list. This network has 1, 2, 3, 4 enabled synapses. One of the synapses has been disabled. This parent has 1, 2, 3, 4, 5, 1, 2, 3, 4, 5 synapses, and 2 disabled synapses. Okay, you can see just by looking at them that they have different cognitive architectures. They have different numbers of neurons and they're wired up in different ways. How can we possibly recombine these two seemingly separate networks? The way that we do this is to take each of these two lists. We try and line up these lists as best we can. We try and line up these lists as both we can, but of course these lists have different lengths. So how do we know which element in which list should be lined up with what other element in whatever other list? We're going to line them up based on their innovation numbers. You'll notice the innovation numbers march from 1 upward in parent 1, and the innovation numbers also march from 1 upward in parent 2. So I'm going to pause here for a moment, and before I talk more about crossover, I want to talk about where this innovation number comes from. So I'm going to back up a slide. Okay. The innovation numbers are historical markings. So what does this mean? What this means is that as the evolutionary algorithm runs, as the NEAT algorithm runs, whenever mutation adds a synapse or a neuron, it's going to give that synapse or neuron a unique identification number It's an integer. And then it's going to increment the global counter so that the next synapse or neuron that is added by mutation to any genome in the population, it's going to get a new number. Okay, so we're going to keep a global counter as neat runs, and every time a neuron or synapse is added by mutation, we're going to assign the value of that global counter and then increment it. This means that early on, every neuron and synapse in the population has its own innovation number. In the very first generation, when we're creating a population of genomes at random, we can guarantee that every neuron and synapse is going to have its own innovation number. So I'm sorry, it's not just when they're added by mutation, but also when we construct these genomes from scratch in the first generation. Everybody gets their own number. But as evolution continues and genomes produce offspring, those offspring inherit the innovation numbers from their parents. The moment that happens, there are neurons and synapses throughout the population of genomes that have the same innovation number. If we have actually, let's stay here for a moment. Let's stay here for a moment. If we have one parent, and that parent crosses with another parent and produces a couple of children, and then the same parent mates with another parent and produces more children, those children are inheriting the innovation numbers from that parent that's been involved in two reproduction events. If you think about a phylogenetic tree or a family tree where you have the founder at the top or the founding parents at the top, and you have children and then more grandchildren and then more great-grandchildren, obviously they are inheriting the genetic material from their ancestors. You can then pick two descendants. We can pick, for example, two grandchildren, and we can find within the genomes of those two grandchildren, we can find genes that have the same innovation number. If we find the same innovation number in two grandchildren, what does that tell us about the ancestor? If you have an idea, go ahead and type it into chat. So if we're looking at the synapses inside two grandchildren, and as we're marching around those two synapse lists, we come across the same innovation number 7, let's say. It appears in both grandchildren. What does that mean? We can be sure that that innovation number of 7 wasn't added to the second grandchild by a mutation, for example, because if it was, that number would be unique. That number from the global counter wouldn't be found anywhere else in the population. So if two individuals have the same innovation number for one of their synapses, what does that mean? Any ideas? It's obviously inherited from the parents, right? Those two synapses might appear at different points in the two synapse list. They don't have to appear at the same position. Innovation number is not position. In mammalian genetics, more or less genes that code for the same things tend to be at more or less the same positions in the chromosomes in the chromosome of mother and the chromosome of father, but that's not always true in nature. So how do we know what pieces to line up? The way we're going to determine which pieces are similar. So going back to the cartoon here, I said that this hidden neuron was computing function A, and the first hidden neuron in parent 1 was computing function A, and the third hidden neuron in the second parent was computing subfunction A. But how do I know that? The only way I would know that is there is some similarity in how these two neurons are behaving. When they receive similar values, they tend to produce similar outputs. They're computing, they're acting in sort of the same way. That's a very difficult thing to detect by looking at the numbers flowing in and out of neurons. An easier way to do this is to identify which neurons or which synapses in two neural networks have the same common ancestor. They have evolved from the parent or grandparent, and by definition, because they've evolved from a common ancestor, they probably do similar things inside their respective neural networks. They may not do exactly the same thing because there may have been some mutations along the way, but this historical marker is a good proxy or a good stand-in for parts of the phenotype that do the same thing. Okay, so if that's true, then we can start to line up synapses based on common innovation numbers. So whatever role synapse 1 performs in parent 1, and actually, let's see, synapse 1 isn't a good example because it's disabled in the second parent. Let's look at synapse 2. It has the same innovation number in parent 1 and parent 2, innovation number 2, which means parent 1 and parent 2 must be distantly related. They must have a common ancestor, and that common ancestor suffered a mutation that added synapse 2. There was one copy of synapse 2 in the ancestor, and the ancestors passed that along to the parents. You can see that it has similar connectivity in both parents. In both cases, it wires up neuron 2 to neuron 4. So that's already a hint that it's probably doing the same thing. But if we look at these two networks, the synapse looks different, right? It's sort of, there's different connectivity. So it's probably similar, but not quite the same. Okay, so we've got these two lists. We start with this element in the list. The first element has an innovation number of 1. We go looking for another element in list 2. We find one that has that number, so we line that up under the first element from the first list. We move on to the second element in the list, synapse 2. It also has a counterpart in parent 2. So we move the second element of the second list down here. Now we come to a third innovation number 3. So we're marching up in innovation numbers. We find that we have a synapse that answers to that innovation number in parent 3. So we put it here. But there is no corresponding allele or no corresponding synapse in parent 1. So we leave that spot blank in parent 1 and we continue. So we are lining up what we think are synapses that do similar things. And to produce the offspring, we now march from left to right along these coupled lists. And at each point, if there are 2 candidate alleles or 2 candidate synapses, we flip a coin. Heads, we copy the synapse from parent 2. Tails, which occurred in this case, we copy the synapse from parent 2 into the offspring. We go to the next element in the paired list. There are 2 copies, so again, we flip a coin. In this case, the coin came up heads, so we copy in the synapse from parent 1. When we get to parent 2, we get to position 3, there is no corresponding synapse in parent 1, so we just copy in the one from the parent that exists. This is called a disjoint gene. It has no corresponding element. Here's a disjoint gene or disjoint synapse in parent 1. We can see that parent 2 encodes a slightly larger network than parent 1. It has 2 additional synapses. These are excess genes. Sorry, I misspoke. In the case where we have a disjoint gene or an excess gene, we again flip a coin. And if we flip the coin's result points to an empty slot, then we move that empty slot into the offspring. Nothing is copied. Otherwise, we copy in the existing synapse. Okay, so we now have this offspring that has a synapse list. It also has a neuron list. We do the same thing we just did with synapses with neurons, but that's not shown in this image. And you notice that when we construct the offspring phenotype from the offspring genotype, it produces a network that looks kind of like parent 2 and kind of like parent 1. There is nothing about this image that tells us whether the offspring network looks like the parent offspring, but there's nothing that tells us that this neural network will compute a similar function to the function computed by parent 1 and parent 2. If parent 1 computes the XOR function and parent 2 also computes the XOR function but in a slightly different way, there's nothing that guarantees that the offspring also produces the XOR function, but in a slightly different way from two parents. We don't know that we've retained the functionality from both parents. Okay, I just said all that. Okay. In the interest of time, I'm not going to show you some results from the NEAT algorithm. You can go and have a look at the paper, which is assigned as reading for today. But take my word for it. It definitely does often produce offspring networks that are as good, if not better, than their parents. There are many times when this random process produces children that are similar to their parents and often perform a little bit better than either parent, because neither parent is perfect, and by the right flips of the coin might bring together the best of two worlds from the two parents. Any questions about that before we switch and start talking about a second algorithm called Hypernate? If you have a longer question and want to unmute yourself and ask it, that's perfectly fine. All good? Okay, so let's move on. Okay. The next algorithm we're going to look at, Hypernate, as the name implies, is going to build an algorithm on top of NEAT. Hypernate is designed to solve a slightly different problem, but I'm not going to talk about that problem until I talk about Hypernate. The second problem is a little bit more subtle, takes a little bit to wrap your mind around it. So let's talk about Hypernate first. Okay. Hypernate is designed, like NEAT, to evolve neural networks. And it's going to evolve a particular kind of neural network called a compositional pattern producing network, or a CPPN, not to be confused with CTRNNs, continuous time recurrent neural networks, which we spent a fair bit of time talking about. CPPNs are very different animals, and they're designed to do something very different. And like CTRNNs, we will unpack what each of the terms in CPPNs mean. Here's a CPPN over here. The inner parts of this network should look very familiar to you. We got 6 neurons, and we have synapses that connect those neurons together. We have two inputs to the network, and we have one output. You'll notice that there are some drawings inside these nodes. We'll come back to that in a moment. CPPNs are designed to take as input not sensor values from a robot or pixels from an image. They're meant to take as input coordinates. So this is a little bit different from anything we've seen before. So I'm going to do a little bit of work at my whiteboard here. You can all see that all right? X and Y. Okay. What we're going to do is we're going to assume that we have an empty two-dimensional space. And for simplicity, we'll assume that X ranges between 0 and 1, and the same thing for Y. We're going to visit regular points within this space. This is a lattice. We're going to pick a resolution because obviously there are an infinite number of points within the unit square here. We're going to pick a particular resolution. For each CPPN in the evolving population, so Hypernate, as always, is an evolutionary algorithm. It starts with a random collection of networks of CPPNs. We're going to take the first CPPN in the population. We are then going to supply a particular coordinate into that CPPN, the X and Y coordinates of that point, and there is a single value that is going to arrive at the output. Okay, so I'm going to start not by using the cartoon CPPN that you see here, but a much simpler CPPN. We're going to have the X input neuron and the Y output neuron, and we're going to have a single output neuron, which I'm going to call B for black. Okay, very, very simple network, and we're going to have a single synapse that connects X to the output, and I'm going to give this synapse a weight of 0, and I'm going to put a diagonal line inside the output neuron, where that diagonal line is meant to represent the activation function. Remember, the activation function takes whatever value is arriving at the neuron and then squashing it or altering that value in some way. There's different ways we can do that. I'm going to start by hopefully you can see that it's a little bit smaller. I've got a small X and a small Y here. So this is just the identity function. This means that whatever value arrives, I'm going to apply the identity activation function, which means I'm not going to alter the weighted sum at all. Whatever value comes in, that's the value that's going out. This is about the simplest possible network you can imagine. Okay, so we're going to start evaluating this CPPN by visiting X equals 0 and Y equals 0. So we have 0 and 0 entering the network. 0 times 1 is 0, and 0 pushed through the identity function is 0. So the amount of B, or the amount of black, that I'm going to place at this particular position is 0. I'm going to place no black at this position. So you can think of the output of a CPPN as painting black into this two-dimensional space. That's why on the slide here there's this cartoon picture of a CPPN and a cartoon picture of a fly. We'll come back to why it's a fly in a moment. But for the moment, we're going to treat Hypernate or these CPPNs as networks that paint pictures in two-dimensional space. Okay, I'm going to move over 1, and I'm going to use a resolution of 0.1. So the next position has an X value of 0.1 and a Y value of 0. If I put 0.1 and 0 through the input layer of the CPPN, what value do I get at the output neuron? This network is simple enough that you should be able to do this in your head. We're feeding in 0.1 and 0 to the input layer. What value do we get at the output layer? Exactly. So we've got 0.1 black. So a little bit of black is going to go at this position. We're going to move along to the next position, X equals 0.2, Y equals 0. You can convince yourself that the CPPN will print out a little bit more black at this point. And as we march along, we are going to start to paint an increasing amount of black along the horizontal axis. Okay, that's just the first row. Now we're going to use the same CPPN and get it to paint the next row. What's the next row going to look like? What's the pattern that the CPPN is going to deposit in this second layer? What's that row going to look like? We're going to feed in, we're going to start at X equals 0, Y equals 0.1. So we're feeding in 0. In this case, we're now feeding in 0 and 0.1. What's the value arriving at the output layer? The value is 0. We are not connecting Y to the output layer. So as Alex says, and as Matt says, the second row is going to look exactly like the first row because the only thing we're changing in the second row is Y, and Y does not influence the output. So we're going to get the same picture. So if I continue this over the entire space, we're going to get a pattern that darkens as we move from the left side of the space to the right side of the space. So that describes the pattern producing part of the CPPN. We'll talk about composition in a moment. Just to help you build up your intuition about CPPNs, let's imagine in this initial random population, there is another CPPN that has the same cognitive architecture, 2 input neurons and 1 output neuron, but this one has a single synapse that connects Y to the output layer and no synapse connecting X to the output layer. What pattern is that CPPN going to produce? We've changed the cognitive architecture, we've modified the CPPN, and it's going to modify the image. As Logan says, in this case, the image is going to get darker as Y increases, but does not change with X. Okay, you got it. So again, to help you build up an intuition about CPPNs, you can do this on your own time. It's fun to think about to do it in the opposite order. Instead of drawing a CPPN and thinking about what image or pattern it produces, come up with a pattern and see if you can write down a CPPN that produces that pattern. Let's imagine the pattern darkens as we move up and to the right in this image. What would be a CPPN? What would be a cognitive architecture for a CPPN that produces that image? Okay, let's talk about the compositional part of CPPNs now for a moment. We are feeding in a coordinate system, and the values of that coordinate system we are computing a function on them, right? We are computing their values and transforming those values. If we have a hidden layer, if we have multiple neurons between the input and the output, then we are composing together a number of functions. We're going to apply this function to the X and Y coordinate system. And if you think about, for example, the 2 values that are coming out of this particular neuron, the 2 values that are coming out here, you can think of them as a transformed coordinate space. If I were to write X and Y on these 2 outgoing connections, and we drew a separate X and Y axes, we are transforming or twisting the coordinate space. So what a CPPN does is to compose coordinate transforms, and that's a little bit beyond what we're going to cover in this class, but for the mathematically inclined students in the class that know about coordinate transforms, you can sort of understand how this works. Okay, let's talk about one other detail of CPPNs. As I mentioned, inside each of these neurons, we can place different activation functions. At the moment, everything we've seen in this class is, again, us as the investigators. We decide on the activation function, like the tan-H function, which you're using in the assignments. And that tan-H function you're assigning to every neuron in the neural network. Again, we're now going to take, in Hypernate, we're taking another step back. We're going to allow evolution to alter the cognitive architecture of these CPPNs. And mutations can also alter or change activation functions inside individual neurons. So the little pictures that you see inside are meant to represent the activation function. The inset axes, the X in the inset axis, represents the raw weighted sum arriving at that network. We go to that X value, and then we move up and find the corresponding Y value in the inset axis, and that's the value that the neuron outputs. You'll be able to see in this picture that there's a Gaussian activation function that tends to produce values near the center. if you work that through, and you can actually play the CPPN game with pen and paper that we were just playing, if you do that, that tends to produce pictures or patterns that are bilaterally symmetric. Stuff on the left side of the image looks the same as the right side of the image. If that Gaussian is applied to the x-axis, that's what you get. If the Gaussian is applied to the incoming y values, then you get the top part of the image tends to look the same as the bottom part of the image. So that's what this cartoon is meant to represent. Our little fly here is mostly bilaterally symmetric. So the Gaussian activation function that you see down here is receiving as input the x value. Actually, in this cartoon, that's not quite true. You can see it's much more complicated than that. But you can make a CPPN where x feeds into a Gaussian activation function rather than the identity activation function, and you'll get a pattern of black that is bilaterally symmetric. The left side of the picture will look the same as the right side of the picture. You'll notice that there are also oscillating activation functions. So in Hypernate, they allow things like applying the sine function to the arriving input to the neuron. Again, you can play the CPPN game and do this on your own. And in this cartoon example here, if we have a neuron and that neuron has the sine activation function, and that neuron receives the y coordinates, it will tend to produce a regular oscillation in the amount of black along the y-axis. By composing these functions together, we can get more than one of these phenomenon in the same image. As you can see in this simple cartoon here, we have bilateral symmetry across the x-axis, and we have oscillating symmetry or repeating patterns along the vertical axis. Finally, they put a little heart in here to show that a CPPN can also paint specific details that are liberated from or independent of these larger patterns. So far, so good? Okay, so why is it called Hypernate? So Hypernate is an algorithm that evolves populations of CPPNs. So CPPNs are a type of neural network, and Hypernate is a type of evolutionary algorithm. Hypernate evolves populations of CPPNs, and Hypernate uses the NEAT algorithm to mutate and recombine the genetic material or the genomes that describe these CPPNs. Okay, so that's how everything fits together. So just to remind you, NEAT is designed to allow evolution to alter the cognitive architecture and the synaptic weights of neural networks. And Hypernate is designed to evolve particular types of neural networks using NEAT that paint regular patterns in space. Okay. I just realized one detail I skipped over was talking about this enable and disable function. Why is that included? A lot of the things we've seen in this class are bio-inspired. They're mechanisms from biomechanics or neuroscience or evolutionary biology that we've built into our algorithms. What biological phenomenon does the enabling and disabling flag in NEAT represent? So this part of the NEAT algorithm that we just talked about is obviously inspired by a biological phenomenon, sexual recombination. What about the enable and disable flag? Mutation. We talked about recombination here. In NEAT, there is also mutation operators. Mutation operators, as I mentioned, can add and remove synapses. Mutations can hit the weight part of a list and change the synaptic weight of a particular synapse. It can also hit the enabled and disabled flags, and mutation can disable a synapse or re-enable a disabled synapse. Any biologists here? So for a long time, there's been a heated debate in biology about the so-called junk DNA, and I should remember to type junk in. Logan said epigenetics. Yes, absolutely. Let's talk about epigenetics in a moment. Good catch. So junk DNA is this idea that there are certain parts of the genome which are not expressed or not often expressed in the organism. And there was an early theory that said junk DNA was just stuff that was left over from before. I'll bet you if you look at your code base now for the assignments, there are parts of your code in which parts of the code are commented out. And those commented out pieces of code are riding along from one branch in your repository to the next. Stuff that's left over from before. That was a common idea early on in evolutionary biology when junk DNA was discovered. But junk DNA usually now is put in quotations, meaning we're not quite sure. It's possible that parts of the DNA that seem inactive are actually still providing some useful function to their host organism. Logan and Toby mentioned epigenetics here. Epigenetics is the idea of stuff that rides along with the genome other than the genes itself that influence gene expression or have some impact on the phenotype. When we talk about the genotype, usually what we're talking about is just the genes, and all the other machinery is there to help turn the genes into phenotype, into fur and feathers and bones and brains and so on. Epigenetics is an exciting branch of evolutionary theory, evolutionary biology, that says there are other things going on that influences or complicates how genes are expressed in the host organism. Again, that's beyond the scope of this course, but for those that are interested, actually this is still an open question in evolutionary algorithms and evolutionary robotics. How useful is epigenetics, and is it useful at all? And junk DNA or allowing mutation to enable and disable genes during evolution, that's one example of an epigenetic mechanism. There are hundreds and probably thousands of other ones that obviously we're not going to have a chance to get to in this course. Okay. So let's come back to Hypernate. Again, we were talking about mutating it. So I'm going to stop drawing now, and I want you to just do these mental exercises in your head. You can imagine a population of CPPNs. Each CPPN produces its own unique pattern. And as evolution proceeds and the neat algorithm mutates or recombines CPPNs into new CPPNs, those new CPPNs produce new patterns. If you create random CPPNs, like we just did at the board here, they tend to produce non-random patterns. If you write down a random CPPN with pen and paper and then draw the pattern that's produced, you don't get white noise. You don't get a random sprinkling of black and gray and white pixels. So what Hypernate is designed to do is to bias evolution, is to bias the neat algorithm or to bias the evolutionary algorithm to a subset of images, which are images that have regular patterns. What do I mean by regular? Regular means things like this. So instead of random noise, you tend to get repeated patterns. You tend to get gradients, things that go from light to dark or dark to light. You get repetition. You get a repetition or variation on a theme. So depending on what else is going on in the CPPN, we can get repetition like the creation of multiple segments. But other parts of the CPPN can modify each segment to produce a unique version of that segment. So the problem that Hypernate was designed to solve is that, as you probably already started to figure out in your assignments, if you create random neural networks, like the ones you have in your robot, it tends to produce random behavior. It takes evolution a while to find a neural network that produces a regular pattern. In your work at the moment, that regular pattern is not regular structure, it's regular function. take a step, take another step, take another step, and take another step. In nature, when there is a random modification through mutation to a genome, it usually has a non-random impact on the child, meaning it doesn't randomly move parts of the child around. It alters the expression of regular patterns. There are mutations in fruit flies that will replace the antenna with legs and vice versa. Sounds like kind of a crazy thing. These are known as homeotic mutants. Again, you can go look that up if you're interested. Mother Nature spent a long time evolving genomes so that when a genome receives a random modification, it produces a non-random change on the phenotype. You can imagine why that is so useful. If a random mutation randomly moved around parts of an organism, most of those random rearrangements are fatal. That's a problem in evolutionary algorithms and evolutionary robotics. We do not want random changes to the genotype to produce random changes to the phenotype. We want it to produce changes, but we want those changes to be non-random, to be alterations in regularity inside the pattern. Okay, it's a bit of a subtle problem, and Hypernate is kind of a subtle fix to this problem. Neat and Hypernate are now very popular algorithms. Neat was introduced back in 2001, so it's the Neat algorithm is now 20 years old. Hypernate is a little bit younger than that. They are still used extensively. We use Neat and Hypernate all the time in my lab. They're very powerful. Not just because they produce regular patterns and they allow an evolutionary algorithm to walk through the space of all regular patterns. They're also very general purpose. So up till now, we've been talking about CPPNs painting pretty pictures, which may or may not be useful to you. But with relatively simple changes, you can get Hypernate to do other things. Let's imagine instead of painting two-dimensional pictures, I wanted CPPNs to produce regular pictures in three-dimensional space. How would I need to change this Hypernate algorithm that I just described to you? How would you change it so that now it paints patterns in three-dimensional space? We add a third input. We add a Z input. And now we have a unit cube, and we visit regular points inside the 3D cube, and we feed in the X, Y, and Z coordinates from that cube, and it tells us how much black paint to place at that point in the cube. You'll see in the cartoon here that the picture is in color rather than in black and white. How would you alter a CPPN to produce colorized images rather than black and white images? Any ideas? Right, so not RGB input, but RGB output, right? So at the moment we have one output neuron, which is B. We delete the B output neuron and add three new output neurons, R, G, and B. So now, whatever we feed in to the input layer, whatever position we're at, there are going to be three values that arrive at the output layer, and they're going to those three floating point values are going to represent the amount of red, green, and blue we should place at that particular point, right? You can probably imagine coding something up like this in Python relatively easily. Instead of black and white images or colorized images, what happens if we wanted the CPPN to produce a movie or an animation? How would you alter a CPPN so that it's going to paint, let's say, 1,000 frames, and you're going to then stitch those frames together to produce an animation? How would you change your CPPN now? This one's a little trickier. Matt's got it. You add to your input layer, you have X and Y. We add a third input neuron called T, which is your time input, as Matt says, right? We can imagine that that's an integer. So to produce the first frame, we're going to feed in the third, we're going to hold the third input neuron at T equals 0. So that neuron is always feeding in the current frame, or F for frame, doesn't really matter. We're going to march through that frame, every position in that frame, and the CPPN is going to paint a picture. Now we're going to set aside that first frame. We're going to take the same CPPN, and now we're going to set the third input neuron, the T, to 1 meaning the second frame. We're going to pass through the image again. Remember that now there is a different input to the CPPN, so it's likely to produce a different image. To continue that until you get to an input of T equals 1,000, and you have 1,000 images, stitch them together, and you have an animation. Hopefully you can start to see how with relatively simple changes to the input and output layers, you can get the CPPN to do different things. Pictures and animation is great, but this is an evolutionary robotics class. So how do we get CPPNs to construct robots? You're going to see between now and the end of the class at least 3 applications of CPPNs to produce different kinds of robots. Just to give you a feel for how this works, I talked at the beginning of the morning about Xenobots, where Xenobots are constructed from different cells. We tell our evolutionary algorithm that it can construct Xenobots from 2 types of cells, frog skin cells and frog heart muscle cells. We tell it that we want it to put those cells together in a 3-dimensional form. How do you think we altered the CPPN to create simulated Xenobots? We want the evolutionary algorithm to put together skin and heart cells into different 3-dimensional shapes. How do you think you need to alter the CPPN to get Hypernate to evolve Xenobots? We're no longer going to be painting black or painting RGB. Any ideas? Imagine we have 3 output neurons, and output neurons always have our floating point values. Imagine if the first value is higher than the other 2 values. We place a frog skin cell. We place a sphere at that position, a skin cell. If the second number, the second floating point value at the output layer is larger than the first or third value, we place a frog heart muscle cell at that position. If the third number is larger than the first or second number, we don't place any cell at that position and we go to the next position. So now we have a CPPN that is painting frog cells inside a unit cube. So we need XYZ at the input layer and skin, heart, nothing at the output layer. Okay. One student in a previous year altered Hypernate to paint music onto a musical sheet. So you move along the sheet of paper, and it paints notes at different vertical positions and at certain horizontal offsets on the sheet of music. You can get CPPN to quote unquote paint music for you if you like. It's a really cool, as you can imagine, very general purpose tool. Okay, we've got just a few minutes left, so we're going to again think about Hypernate by applying it to a different problem. And this was reported in a more recent paper, so we're going to be talking about this paper for a moment. This is going to seem a little odd at the beginning. We're going to construct a neural network, and then we're going to get Hypernate to evolve neural networks, to evolve CPPNs that paint weights onto this second neural network. So we have a CPPN. It's no longer painting black or color or frog cells. It's going to paint weights. It's going to paint floating point values onto synapses inside a second empty neural network. Okay, seems like kind of an odd thing to do. I'm going to describe it first, and then probably next time we'll see why this is useful. In order to do this, we need to embed the neural network that we're going to paint. We need to embed it into a coordinate system, right? Because remember, the CPPN is always painting in 2D or 3D. It's painting into some space. It could be spatiotemporal. So when we were making our animations, our coordinate system was X, Y, and T. In this experiment here, we're going to have, let's see, maybe I can just skip ahead to show you the robot. So we're gonna have a simple quadruped. It's got 4 legs and a body, as you see here. We're going to embed the neural network that controls this quadruped. We're going to embed this neural network in the 3-dimensional space that you see here. The input neurons, there's going to be a total of 20 of them. We're embedding that in x equals 0 in a 2-dimensional plane. We have 20 hidden neurons, which is embedded in the 2-dimensional plane at x equals 1. And finally, we have 1, 2, 3, 1, 2, 3, 4. 3 times 4 is 12. We have a total of 12 output neurons that is embedded at x equals 2. So we have the input layer, hidden layer, output layer. Okay, so this is x equals 1, x equals 0, x equals 1, x equals 2, and we have y-axis and z-axis. So each neuron has its own 3-dimensional coordinates inside a unit sphere, inside a unit cube. Okay. The neural network that controls this robot has 20 sensor neurons. We have the first, second, third, and fourth leg. Inside each leg, let's take leg 1. Leg 1 has a hip forward and backward proprioceptive sensor, hip forward and backward. The second leg also has a hip forward and backward sensor. Same thing with the third leg, fourth leg. Each of the 4 hips at each of the 4 legs also has its own inward and outward rotation. So the hip, if you like, of each of the 4 legs can rotate in and out, IO, and forward and back, FB. Each leg also has a knee, and there's a third proprioceptive sensor in the knee, reporting the angle of the knee at each point in time. T is simply the touch. T is the 4 touch sensors in the 4 feet. They provide an additional 3 sensors that report the pitch, how much the robot is pitching forward and back, roll, how much the robot is rolling about its long axis, and yaw, how much the robot is rotating left and right as it moves around. And finally, they're feeding in a sinusoidal pattern in this 20th input neuron. What aspect of biology is this sinusoidal neuron representing? We saw this concept last week, I think, when we talked about biomechanics. Why feed a sine wave into a neural network that's controlling a robot? Any ideas? Where have we seen that idea before? Of a network that's receiving a regular oscillatory pattern as input. It's for oscillatory and periodic activation, as Logan mentions. That's right. So it's going to help to bias evolution towards gates for the robot that produce oscillatory motion. You also have neural circuits in your spine that help bias you to produce regular oscillatory patterns. What is that circuit called? In this case, they're representing this neural circuit found in animal spines. They're representing it by a single neuron. Anybody remember what they were called? So these are central pattern generators, or CPGs. You have them in your spine that when they receive a signal from the brain, a non-oscillatory signal from the brain, they receive a command from the brain, they start spontaneously producing sinusoidal patterns. When a chicken's head is cut off, there is an electrical pattern that reaches the spinal cord that produces walking. Okay. That's the input layer. Each input neuron is attached to each hidden layer. So you can imagine the synapses are not drawn here, but you can imagine dense connectivity coming from the x equals 0 layer to the x equals 1 layer. And then there are a set of synaptic weights that go from the hidden layer to the output layer, where we have it's a little bit occluded here. For each of the 4 legs, which corresponds to the four legs. Each leg has 3 output neurons assigned to it. The first output neuron controls the desired angle for forward and back. The second output neuron dictates the desired angle for the hip swinging inward and outward. And the third output neuron is the desired angle for the knee of that leg. So we have 3 motors with 3 motor neurons per leg, 4 legs, 12 output neurons. Okay, so each neuron has a 3-dimensional position. And if you imagine a synapse, for example, that connects this neuron to this neuron, that synapse, if we take the midpoint of that synapse, it also has a 3D location. Okay, so that's the neural network that controls the robot. But we have our population of CPPNs. We take out our first CPPN, and we're going to use that CPPN to paint weights on this neural network. We're going to allow that neural network to control the robot in the simulator. We're going to record the amount of fitness, how far the robot travels, and that number, fitness, is going to be assigned back to the CPPN that painted the neural network controlling the robot. Okay. Again, it takes a little bit to wrap your mind around this. We have 2 separate neural networks. How are we going? What is the input layer and the output layer of the CPPN look like? What should the input neurons be, and what should the output neurons be? We want to construct CPPNs that are able to paint weights onto these synapses. What would you feed as input, and what would you take as output? Remember that the evolutionary algorithm, or NEAT, is going to build and evolve the cognitive architecture between the input layer and the output layer. But we, as the investigators, still need to tell HyperNEAT what goes into the network and what comes out of the network. How would you construct a CPPN to paint synaptic weights onto this neural network, which is embedded in a 3-dimensional space? X, Y as input, weight as output. Okay, let's try that. So we have 2 inputs and 1 output. X and Y, so remember that X indicates which of these 3 layers we're at. So X could be here. Actually, sorry, X is 0 through 1. So X is going to give us some position between the input layer and the output layer, and Y is going to give us some position along the horizontal axis here. Any ideas? Remember, we want to paint weights onto the synapses. So before, in the cartoon example here, we were just picking some random, not random, we were picking a lattice. We were visiting regular points in this 3-dimensional space. In this case, we are not going to do so. We're going to hop from each synapse to the next one. If we arrive at the midpoint of each synapse, what do we get? We're going to place a single number. So Matt's got half of this right. We want one output neuron, where that output neuron is dictating the weight of that synapse. What do we need to feed as input? If we're visiting a synapse, what can you tell me about the midpoint of that synapse? So for example, if we think about this synapse here, here's the midpoint of that synapse. If we imagine the synapse that goes from this neuron to this neuron, you would draw this synapse and it would have a midpoint around here. Okay, we're out of time, so I will leave that for you to think about, and we will come back to this on Tuesday. You have a quiz due tonight. You're working on assignment 9. I wish you a good rest of your day, good weekend. See you back here next week. Bye-bye, everyone.